Hello, AP Bio. Welcome to our video lecture for Chapter 22, The Origin of Species. So this chapter tries to help answer the question, and it's a very difficult question, but the question of how do I go from one population that's one species to two separate populations that evolve into two separate species? And that's obviously a very difficult question. It's not a question that we can answer like with one nice, concise sentence. But one thing that has to happen, and what this chapter is getting at, is for two species to evolve into separate species, or for one species to evolve into separate species, they have to be genetically isolated. And let me give you an example of what I mean by that. So let's say you have a population of mice, and the mice are dark in color. And let's say there's a random mutation that creates a light-colored mouse. And let's just say that the mice are living on light-colored sand. So a light-colored mouse is more fit than the dark ones because it blends in. How does that mutation spread throughout the population, right? Well, it's not horizontal spread. It's not from mouse to mouse. It spreads through reproduction. It spreads through parents giving it to descendants. So that mouse has offspring that have offspring. They mate. They have offspring. Come back in, gosh, say 10 years, and you're going to have probably the whole population be the light-colored mouse. Okay, now, let's say that I have a separate population of mice in that same area that for whatever reason, they can't mate with the original population. We're going to go through reasons why that would be in this chapter. But let's say that they can't mate. So they're isolated genetically. So that light-colored gene, the gene that gives you the light-colored mouse, can't get to that population. And let's say, just to go further, let's say that population finds some darker sand to live on. All right, come back in 10 years, you're going to have two separate populations, a light color population, a dark color population, in different areas that are not mating back and forth. So they're basically, their gene pools are separate. If their gene pools are separate, mutations can't go back and forth between them. Has that created a brand new species or two species? Probably not yet. But one thing that has to happen to create new species is they have to be isolated. You know, if you're swapping genes back and forth, you're going to be one species. The definition of species is you can reproduce fertile offspring. So this chapter goes through a bunch of different mechanisms that create new species by isolating populations. The picture I've chosen for this chapter, this is from the chapter. So this is a, I mean, it's kind of funny in a tragic way. These are two snails trying to mate, all right? And the arrows are pointing to their genital openings, okay? And if you notice, one snail's shell, the pattern's clockwise, and one pattern's counterclockwise. And based upon the shell's pattern dictates, in this case, that the genital openings cannot overlap. So these two snails can mate all day long, or they can try to, but they're not going to create offspring because mechanically the parts don't fit. So this is a case where I've isolated a population. They're actually not isolated. They're in the same place, but they're isolated genetically because they can't have offspring. So this is one way to go from one population of one species to two separate populations that become separate species. Couple definitions really quick. Speciation is just the fancy word for creating new species. Microevolution and macroevolution, we defined these in previous chapters. Microevolution is when you're looking at allele frequencies. This was Hardy-Weinberg. Macroevolution was basically chapter 19 and 20, looking at broad patterns of evolution, as in, you know, what Darwin was doing. There's different ways to define what a species is. The one that we usually use is the biological species concept, which basically says if you're in the same species, you have the potential to breed and to mate and to have offspring which are fertile. So if you're in the same species, you can reproduce offspring that are fertile. This is the one we usually go with. There are some drawbacks to this kind. One, how do you classify species that reproduce asexually, like bacteria? That wouldn't work for this. And how do you classify species that are extinct, where you just have the fossil record? You obviously can't see fossils mate. So there are some other concepts. The morphological species concept is defining species by structural similarities. This is how you would use bacteria or other things that reproduce asexually. Okay, so what we're going to do is we're going to go through different ways to isolate two populations reproductively or genetically. It could be as simple as there's a mountain range between two populations. That'll do it. But it might be something like the issue with the snails that we had on the first slide. The word hybrid, I'm sure you know, the word hybrid just means if you have offspring between two separate species. And what we're going to do, so this chart's from your book. This is a great chart. This goes through all the different sort of blocks that prevent successful reproduction. And notice they're divided into prezygotic and postzygotic. A zygote means you had reproduction, you created a zygote. Prezygotic means you prevented that. Postzygotic means you created an embryo, but for whatever reason it did not survive. You can see on the left here, these are before a mating attempt. These are between mating and fertilization, and these are after fertilization. So we're going to go through each of these, explain what it is, and give an example. So we're going to start with habitat isolation. This is probably the easiest one. This just means the two species don't encounter each other. They live in different habitats. If you live in different habitats, you're probably not reproducing. Temporal isolation is if you could be in the same place, but maybe the mating seasons are different. You breed at different times of the year. If it's plants, you could have a case where one population, the flowers produce pollen in April, and in nearby plants, the flowers, the ovules are ready in July. If the times of the mating seasons don't overlap, obviously you can't reproduce even if you're in the same place. That's temporal isolation. Behavioral isolation, here we have some blue-footed boobies, right? So this would be courtship rituals or whatever that prevent two species from, or prevent members of possibly the same species from reproducing. This is the male doing the dance. This is the female. She's kind of giving him the side eye, like she doesn't want to look at him. She's probably not interested, to be honest. Maybe his dance isn't doing it for her. Okay, yeah, enough of that one. Okay, so those three were prezygotic barriers, right? So behavioral isolation is when you have species that have courtship rituals or behaviors. Here we have two blue-footed boobies. This is the male right here doing the dance. Here's the female. He's looking her in the eye. She's kind of looking off to the side, maybe giving him some side eye. I'm not sure that she's convinced by his dance. But if you have sort of behavioral or courtship rituals where the female is picking which one to mate with, given enough time, that could produce two separate species. So in review, those three were prezygotic barriers: habitat isolation, temporal isolation, and behavioral isolation. So at this point, we're going to have the actual mating attempt. So things that would block fertilization after mating. So this is back to our snail example. So mechanical isolation is when you have morphological differences that prevent mating, like the case where the snail shell, one's counterclockwise, one's clockwise, so the parts literally don't fit. Gametic isolation, this picture is sea urchins. And this could be a case where you have mating, the egg and sperm come together, but for whatever reason, maybe the cell membrane of the egg isn't compatible with the membrane of the sperm, and fertilization doesn't actually happen. Gametes are egg and sperm. So gametic isolation just means the egg and the sperm are not compatible, even if they come in contact with one another. Kids always think this picture is showing fireworks. They're not fireworks. They're very colorful sea urchins. Okay, so to review, these were the prezygotic barriers after mating, but before fertilization. So we've done these 5 right here. We only have 3 more to do. So postzygotic barriers would be ones where you actually create a zygote, possibly even an embryo, but for whatever reason it's not viable. Reduced hybrid viability is the term for, say that you create a zygote. Remember back in chapter, I think it was 16, we were discussing all the developmental cues in the egg and all the genetically coded for cytoplasmic determinants, all those things. What if the egg, the determinants in the egg just aren't compatible with the sperm, such that you do create a zygote, but it doesn't develop right, and eventually the embryo just dies. That's called reduced hybrid viability. You created a hybrid, but it did not survive. Reduced hybrid fertility, this is a picture of a mule. So this is the case where you do create hybrids and they're vigorous, they survive, but they're sterile. So a horse and a donkey can reproduce and they make mules, but mules are sterile. The only ways of making a mule is to go back to a horse and a donkey. So did that create a new species? You know, this is sort of like the asterisk next to species, because, you know, the definition was, if you're the same species, you can produce offspring that are fertile. So this doesn't meet the offspring that are fertile part of the definition. So a horse and donkey are still different species because they don't produce fertile offspring. Hybrid breakdown would be an example where, say your first generation of hybrids are vigorous, they're fertile, but the next generation doesn't survive. Either the hybrids reproduce with each other, or they try and reproduce with a member of the parent species, like a mule trying to reproduce with a horse, such that for every generation, the new generations become feeble or they're sterile, such that the line just sort of dies out. This is like the last. If you get past this block, you've successfully made it. You're in the same species. Okay, so what you need to know from this chapter, so these 8 things, going from being in different habitats to just creating hybrids that eventually break down in subsequent generations, these are the ways of isolating a population genetically because they can't successfully reproduce. If 2 populations can reproduce successfully, they're not going to become different species because they're swapping genes back and forth between their gene pools. As they create descendants, the genes are going back and forth between the gene pools. So these terms, allopatric speciation and sympatric speciation, this is a super important concept. AP exam loves this concept. So allopatric, the root allo means other. The root patric means country. It's the same root that the word patriotic comes from. So the word means other country. So this would be speciation where the animals or whatever they are in different places. Sympatric speciation, sim means the same or together. That would be where they're in the same environment. So let's go through some examples. So allopatric, a river could separate them. One could be on an island, a canyon, you have a mountain form between them, where the 2 populations don't encounter each other anymore. An example, so this is showing a mosquito fish in the Bahamas, and based upon what pond they are in, the predators could be different, and there's different body shapes. There's this sort of thicker body shape and then the more streamlined body shape. This body shape is better for rapid bursts of speed, which if you have a predator, that might be beneficial. This body shape is better for steady swimming, which if there's no predator, being able to swim longer, steadier might get to the food source better. Basically, fish in ponds with predators look like this. Fish in ponds with no predators look like this. And obviously they're in different ponds, so they're isolated. This just shows these are lobsters. So the Isthmus of Panama here that separates the Atlantic from the Pacific, that used to be underwater. So here I have, these 4 different species of lobster or crayfish or whatever they are. I'm sure the book tells you what they are. Used to be one population because they could swim back and forth, because this is not a very, this is a very narrow isthmus, right? When sea level went down and they got separated, they started evolving into separate species because they couldn't swap genes back and forth. So both this example and the one before are examples of allopatric speciation. They're in different places. So for sympatric, this is actually, it's kind of a combo of both. So this is a case study from the book. This is a fascinating case study where scientists were growing fruit flies on when you grow fruit flies, you have to give them a medium, some kind of sugar medium to grow on. And they're growing fruit flies in a medium. And then they separate the flies, and they have some flies that grow on a starch medium and some flies that grow on a maltose medium. And they do this for 40 generations. Where the flies are they're in different jars, they're growing on different mediums. Notice for each, this is important, they have 2 jars for starch and 2 jars for maltose. That's going to serve as the control. Why they do that, we'll see on the next slide. But what's interesting, then after 40 generations of mating, they put them back on the initial medium, which I think was some kind of corn-based medium. It wasn't starch or maltose. And they observed which flies mated with which. They're back in the same place. They can mate. And look at the data. Females that had been grown, like they hadn't, their ancestors had been grown on a starch medium, and males mated 22 times. Maltose and maltose mated 20 times. Maltose female and maltose female and starch male only mated 9 times. Starch females and maltose males only mated 8 times. So it's like the flies have a preference for what medium their ancestors were grown on. Now, why I even know, I have no clue. Somehow genetically, some kind of epigenetics happened where the genes changed or the genes didn't change, but how like if they were methylated or whatnot changed due to what medium they were growing on. But clearly, like this is sympatric because they're in the same place, but they have a preference. Now the control, so maybe the scientists were like, maybe because they were in different jars, maybe that made a difference. So they had, like this is starch population 1. So this is a female and a male that were on starch population 1 18 times. This female was grown in starch population 2. This was grown in starch population 1. This male, they made it 15 times. So here, like, look at the numbers. It doesn't really make a difference as to what jar they were in. They could have been in different jars. What makes a difference is, was the jar starch or was the jar maltose? So you should understand why this is the control, because it makes sure that the preference is due to what medium they were on and not what jar they were in. Being in different jars didn't make a difference. This just defines sympatric. This is speciation that takes place when you're in the same geographic area, like the case with the fruit flies when they put them back on the corn-based medium. So last thing in this chapter, so a big question in evolution, and this is, I don't know, this is kind of a silly question, but whatever, is how long does it take for species to evolve? And I mean, the answer is it takes at least 2 generations. 2 generations is the minimum, because you have to have at least 1 generation of offspring to see evolution. These 2 terms, punctuated equilibrium and the gradualism. So punctuated equilibrium, look at the picture. Here I had rapid change really quick and then no change at all. So it's punctuated, like it stopped, it started, and it stopped, like at the period of a sentence. The gradual model shows gradual change, where we have like intermediates over time. Punctuated equilibrium, again, is the view that evolutionary change happens in a rapid burst and then nothing. Gradualism says that it's slow, steady change over time. Which one happens? Well, the answer is both of them happen. What do you think really dictates how fast evolution happens? Well, there's 2 things. One, nature dictates it, because nature is what's choosing the selective pressures on a species. Remember, but the first slide when we had the light mice and the dark mice, when they're on different sand colors, nature's pushing them in different directions. Selection's pushing this population to be darker and this one to be lighter. So nature dictates evolution, but also just what mutations you have. Remember, the mutations is largely random. So there's a random element to it, but there's also an element to it that's driven by nature, by natural selection. All right, that's the end. That's enough for one chapter. Hope that was helpful. I will see you guys next time.